Okay, so good afternoon, everyone, and welcome to the plenary colloquium of the Italian Society of Statistical Physics. So today we are really very happy to have with us Miguel Angel Muñoz for the University of Granada. So I think you all know Miguel very well, but let me say a few words about him. He's a professor of the University of Granada, where he's also the director of the Institute of Theoretical and Computational Physics at the moment. And Miguel is an expert on criticality and scaling in equilibrium and non-equilibrium statistical physics in general, but in particular in living system. And you might know that on this subject, he has written a review that is really a reference in the field. And of course, one of his main expertise is the application of statistical physics method to the brain. And today he is going to talk about some very recent and important result he obtained on this subject with his colleague in Granada. So Miguel, thank you again, and I leave the floor to you, so please.
Thank you very, very much. Thank you, Rafaela, thank you, Roberto, and all the people at the SIFS for inviting me. It's really a pleasure and a privilege to be here with you, well, here, there, I don't know, with you. And I really feel that I don't really feel a foreigner in this community, even if I'm Spaniard. But I really feel from the physics point of view, or statistical physics point of view, I really feel Italian in my heart. So I have much to owe to the Italian community for all the things I learned, as you will see in my very first slide. So I'm going to, the title is a bit long, so don't worry, I will talk about brain activity, criticality, and I will try to connect the actual brain with some issues with artificial intelligence, hopefully. Okay, so the starting point is this observation that the cerebral cortex is always in a state of ongoing activity. And this takes a lot of energy from us. It's like 20% of our energy goes to keep this thing on. So one should understand what's the functionality for this. The very first time I heard about this was to Daniel Amit, and I met him in Rome, I don't know how many years ago, many years ago. And for some reason that when I arrived, we became friends and he took me to drink wine and things in Rome. And one of the first conversations, he told me that he was not interested in statistical field theory anymore, that he was very much into brain. And he told me about the experiments they were conducting with the monkeys and this thing that was puzzling him very much of the continuous activity of the neurons and its functionality. And he was developing these models with studying that was after Hopfield model and so on. But anyway, that's just to say that the origin of this research goes back 25 plus years ago to Daniel Amit in Rome. So as physicists, we would like to characterize this activity, the statistical properties. And we would like very much to understand that as the result of a physical process. Okay, we want to do theory, we want to have a model. And then we want to understand what's that activity useful for. And if we learn something of why this works the way it works, maybe we can extract some lesson to develop new prototypes of artificial intelligence. Okay, that's the goal. So my research is, I'm a theoretician, but here we are going to be very data-driven. So we want to stick to what we see in the brain, not just construct very metaphorical models, but we want to keep ourselves constrained by data, right? And the nice thing is that our friends, experimentalists in the last decades, have developed very nice and sophisticated strategies to detect this brain activity. And this is very, very, very rich field, but just very, very simplistic. I differentiate here between the macro scale, so the full brain, where you have techniques like this. For example, well, EEG, you all know, or MEG, magnetoencephalography, you know. This is fMRI, so functional magnetoresonance imaging, fMRI from now on, and where you put the person in the scan and you see patterns like this. So you have a number of regions where you detect activity, and you have time series here, and you see, for example, the yellow corresponding to some region, let's say here, and the orange corresponding to another region, let's say here. they seem to be highly correlated. So they fluctuate together. They go up and down together. Instead, the bluish seems to be kind of anti-correlated, right? So the activity, first observation, is not random. There is some structure here, and we would like to understand that structure, right? Similarly, well, I will discuss also this diffusion tensor imaging that allows to measure not only the activity, but also the structure, the map of connections. I will show you this in a few minutes. And at the micro scale, when you look at actual neurons, this is rather amazing. Maybe some of you are not aware that with these optogenetic techniques, they are able to look at the microscope individual neurons, and they change color, they become fluorescent whenever they become active. So you have really a movie of neurons getting on and off, and you can track the activity and you can follow what's going on. And you also have these neuropixel probes that you put in the head of the mouse, not in the humans. That's not approved, luckily. And you detect the activity of tens of thousands of neurons at the same time. You can reconstruct time series in very diverse places. Here you have a section of the brain. This is the proof. And here the different regions where you are detecting activity and tracking what neurons are doing as a function of time. And now this is the new world record. I saw this year. We have this light bed microscopy that, to be honest with you, I don't know what it is. I have not investigated, but with this, it seems you can detect the activity of 1 million neurons at the same time. So that's fantastic for a statistical physicist. That's a very nice playground. It's not the Avogadro's number, but I mean, we are growing and we are getting things. So the main focus of my talk will be on microscopic, so actual neurons and actual circuits and how they operate. But just as an introduction, just to give you the flavor, I will discuss first some properties of the large scale brain, of macroscopic analysis, which is much easier to grasp because we all see the brain and we recognize it. So as I was saying before, you can detect with this fMRI the activity at different points, at different locations, right? But you can also detect with this DTA, diffusion tensor imaging, and other technologies, tractography, you can detect the map of connections. So you can say here in this matrix, we are going already to a matrix form. You put here all the regions of interest you have identified, like 1,000, just to have some parcellation of the brain, 1,000, and then you here we are using a color code to see which region is connected to which one, right? And you have these funny matrices where you can detect just by naked eye that there is some structure, right? And then when you measure the correlations or the covariances, for example, during the same time window, you detect here, again, you see that there is some nice structure, but across time, things changing because obviously the person is doing different things, maybe it's changing a state or doing something else. So this is called, in the jargon, the structural connectome. This is called the functional connectome. Now fancy people talk about chronectome to give an idea of time, the chronectome, how this thing is changing. Okay, anyway, as physicists, we would like to have something like this. I give you the structure, the map of connections, and I put here my physical model that I would like to be as simple as possible, but not simpler. And I would like to detect the maps of correlations and also the dynamics of these correlations as I plotted before. That would be our dream, right? As modelers. And the picture is very similar to what we do in physics or in quantum mechanics. You have some structure and you look what are the normal modes or the first excited states. So probably the very simple idea is that the brain should be in some basic resting state that can be perturbed, and then you have some modes that are perturbing that. So it's the same thing that we do in physics. You can have a 1D object or a 2D object or a 3D, and you have these main first modes of vibration. But we do something very similar with the brain structure. So we take the network of connections of the brain, and we look for the first modes of oscillation. The first one is constant, which is just switching on and off. Second one is division in hemispheres. The third one is propagating, is something activating the occipital and frontal part alternatively. And here you have a collection of some of the first 20 eigenmodes, which are eigenvectors of the Laplacian defined on this structure, right? The very same thing we do in physics. This is actually the brain of a PhD student analyzed in Bilbao, our collaborator Jesus Cortes at Las Biocruces. They have a fantastic group of neuroimaging, and essentially most of the data we analyze come from them or from the Human Connectome project that they also participate in, and they share data, right? So this is activity in real time, and you have regions that are switching on and off, as you see. And here is the projection in 4 nodes that you see that from time to time, the first mode gets on, and the weight is changing in the different modes, very similar to what type of analysis we do in physics, right? The funny thing is that now I'm not plotting just 4 modes, and now I'm plotting 10 to the 6 to 10 to the 7 modes, a lot of them. And what I see is that in real time, they are getting on and off in a very interesting way. Observe that this is a log-log scale. So we have a power law of activation of modes, of these eigenmodes that we have defined in the structure of the brain. This is very, very nice. And also I will be talking about the covariance matrix, which is an object I like. So again, imagine here I have my 1000 something regions also here, and this matrix is color coding the level of correlations, right? Like in this plot I showed you before, some parts are more correlated, some are less correlated or anti-correlated. So I have this matrix, and I can diagonalize it. This is the type of thing you do when you do principal component analysis, right? That you diagonalize and look in the principal directions. You conclude that in the principal direction, the activity is changing. So if you put here the spectrum of this covariance matrix, what you see again is that this is a power law. Okay, so it's not that you have a few modes. Many people talking about this functional magnetoresonance imaging, they talk about 6, 7, 8 modes or networks, they call them, of activation in the brain, and they have names and they correspond to different things. But just isolating 7 is, I go here 1, 2, 3, 4, 5, 6, 7 is like cutting the information here. You could equally say that you have 14 and cut here or there. You have a whole hierarchy of covariance within covariance and covariance and covariance. You can represent this with a dendrogram of who is connected, who is correlated with whom, and you have also, again, here you can appreciate this nested hierarchy of covariances. So a very complex, very fractal-like structure of covariances, right? So again, well, this is a simple picture. When you do principal component analysis, the thing is very simple. Here you have a 3-dimensional space, but when you project in the 2 main directions of the covariance matrix, you can have in this example that just with 2 of them, you can do the whole thing. You can do a dimensional reduction. Okay, you are lower dimensional embedding of your data. We would like to do something like that in the brain because it's very high dimensional, but one has to be very careful because you can truncate it and do very uncontrolled approximations on things. And of course, when I see this power law in the covariance matrix, I, of course, as a statistical physicist, I think of the Ising model. Of course, how couldn't I, right? So you take Ising and you see that in the disorder phase, you have configurations like this. And if you look at the power spectrum, what you see is that is wise noise. All modes are equally relevant. So if you diagonalize your covariance matrix here, you see something flat, a lot of modes, but all equally stupidly relevant, is noise. In the order phase, you have one dominant mode, which is the collective mode that has broken the symmetry, right? Here is black, and then just noise, which is again flat. But if you go to critical and you are here, you have correlations at all the scales. And this is the fractal. And when you look the covariance matrix, you diagonalize the covariance matrix, you have this. This is the propagator in physics, and it's a power law. So it's the same thing that you see. I mean, it has the same qualitative shape that you see in the data in the brain, right? And of course, we know this is the indication of a scale invariance, as you see in this beautiful video by this person here, in which you are doing actually the Kadanov coarse grains in real space, and you see the scale invariance. I put this because afterwards we will try to do some renormalization with brain data, doing the same thing that we are doing here, right? I just show you some numerical work, because I said you before that we would like to model this. So this is unpublished results. We have our model that I'm not showing to you. It's a model with excitation, inhibition, the network connection, and something relatively simple. And we would like to reproduce this. This is the empirical data I showed you before, showing, for example, how the activity projects in the basis of eigenvector of the Laplacian. And these are our simulated data, right? So this is real and this is simulated, and we get something, I mean, very similar. I wouldn't say identical, it's work in progress, but we get something very, very similar. And also for the covariance matrix, we get, this is the real data and this is our model. But this works only when we fit our model, whatever it is that I have not shown it to you, but we have to tune it close to a critical point. So very similar to Ising, we have to go to criticality. Actually, we discuss always also this concept of Griffith's phases that our group introduced in neuroscience, and that comes from condensed matter physics, and tells you that sometimes when you have disorder, when you have heterogeneity, there is not a critical point, but a critical region. So that's sometimes useful for these problems. But anyway, I will just talk about critical. And this is a PRL paper that I'm very proud and fond of, because just appeared 1 week ago. It's also featured in these things. And we introduced a new method, not just to say that the reality is close to criticality because it resembles the models, but we can actually measure it. We have developed a technique to measure how far from criticality is our actual data from brain. But I won't stop much here. Or just to illustrate this new method, I showed you this. These are healthy people, and these are frequency bands in the brain here. And what this blue line here is critical. And you see that we are quite close to critical in this alpha band, which is important for the brain, but not in other bands which are less important. Instead, if you take people with Parkinson's disease, you see that the distance to criticality is very different. A lot of modes that shouldn't be there are close to critical, so are closer to becoming unstable, and that's why the Parkinson's patient is experiencing something unpleasant, right? Anyway, so people, of course, including myself, and a very large community that includes, of course, Lucilla, who is in the audience, and Silvia, and many others Italian colleagues, where we started trying to understand why. I mean, the model works better if it's critical, okay, but why? So maybe because critical allows for large correlations and long time scales and responsivity, many possible different things. So I wrote this review paper already almost 5 years ago, my God, right? In which, among other things, I discussed this neural activity, but also similar principles acting on gene regulatory networks, or in the collective behavior of cells, collective motion, one of the favorite topics of Giorgio Parisi, Cavagna, Irene Giardina, etc. Right? And there are very interesting works about these functional benefits of criticality. And now it's very fashionable to try to see, maybe I show you this. So it seems that the good state is close to critical, and if you are too much critical or you go beyond the limit of criticality, it's associated to different pathological state. Like if you take psychedelic, you take LSD acid, you come over the limit of instability, and weird things happen. And if you are in anesthesia, sedation, deep sleep, or some other problems, you have a reduced level of consciousness, or you are further away from criticality. Anyway, so that was an introduction, maybe a longish one, because as I said before, what I wanted to talk really is about the microscopic level, the real actual neurons, and okay, let me go ahead. So as I said, we have these recordings with tens of thousands of them. So we are very happy we can look these individual neurons. And I have to start presenting this work by the group of Bialek, who 5 years ago introduced this phenomenological renormalization group approach to study this data, these very rich data sets. So what they did is very simple, is that you take time series from real neurons, like these ones here. This is a sketch, obviously. So you have different neurons, 8 here, and this time. And what you do is a cross-graining in the same spirit as you do in the renormalization group, in the Kalanov approximation, in the Kalanov spirit. You put together the activity of this and this, you sum them, and maybe you normalize it with some constants. That's not very important for us now. And you do it pairwise. Here you don't have really an idea of proximity. I mean, here in easy model, you know who is neighbor of who. But neurons, they have long range connections. They have these synaptic and dendrites connections. So it's very difficult to know exactly who is neighbor with whom. So what they did is to establish a criterion of correlation. You put together the neurons that act more correlated, right? And you do this in a hierarchical way. I mean, and you have a new level of now super neurons, grouping pairs of them, and you iterate, and you have now super neurons and all the way. So the same thing that you do in the RG, many neurons, and less, and so on progressively. Okay, and now you track a number of observables, of quantities, and this one, for example, here is the distribution of activity. So you look in one of these clusters here, and you look at the neurons. I mean, some of the clusters that you defined are more active, some are less, and you have some activity that varies. And you have this distribution, which is not Gaussian, and is very scale invariant, because these different colors correspond to the activity at different levels of coarse graining. And the distribution remains amazingly, in my opinion, amazingly invariant, and it's not a Gaussian, so it's not trivial. Here is another property. Whatever. This one is the variance within each of the clusters. Sometimes you have more activity, sometimes less. So you study the variance of the activity as a function of the level of coarsening, right? And what you see is this perfect scaling again, a very nice scaling, which is not one that is what you could get if the neurons were randomly placed, and it's not two that could be the result you would get if they would perfectly correlated. So this one is a central limit theorem, and this one is perfect correlation, and you get something with a very nice scaling, but it's in between, so it's non-trivial. And something similar for temporal scaling, and also for the covariance matrix. So in the same data, you look the covariance matrix, which is one of my favorite objects, and you plot it, and you see that you have very nice collapse. It obeys finite size scaling indeed, because all the different coarse grain levels have different size. So you can, this is a finite size scaling collapse, and it's also beautiful, I think. So you can measure this critical exponent, beta, alpha, z, mu, etc., etc., etc. This is again. So the questions that we tackled with my collaborators, mostly here in Granada, are these ones. I'm gonna try to answer these first 3 questions. First, we have observed this scaling, and these results from the group of Bialek were done in the hippocampus because they wanted to study grid cell and place cells and the role of them, and etc., etc., etc. So our question was, if you look at different regions, could you get something similar, or does this behavior change across regions? That would be a signature of universality. Then we have seen that there is a scale invariance, but is it critical? Is there a phase transition? Is separating different phases? Can we say that? And then, of course, can we model this? So this is my program for the next slides. First of one, universality. So together with Guille, who is my ex-PhD student, now postdoc, still here in Granada, but looking for a new postdoc. He's very good student. So we went to look data of Allen Institute and this paper here in Nature. And we found 16 regions in the mouse brain for which we have enough data as to repeat this type of renormalization group analysis. And here is a summary of results. So look, we have 16 colors for 16 regions. That is not important now which region is which, right? And this is the distribution of activity. And you see that the scaling is amazing across regions. Here, the discrepancies you see are in the tail, and it is still very narrow in the order of -3. So in the bulk, it's very large. And I think that we have a lot of experimental errors. So these results is amazing, I would say. And this is for all regions. And if you look one specific region, you see again that it obeys scaling, finite size scaling. So you put the different sizes and rescale, and you see it beautifully here collapsing. And if you look at the variance, again, it's a non-trivial exponent, 138, same one that they measured, that VLEC measured within error bars, which is between 1 random and fully correlated value 2. And same thing for the other exponents. This is the dynamic, etc., etc. So our conclusion is that we observe a scaling variance across the brain in the mouse. We have very good scaling, I would say, but there is some dispersion in the exponent values. It's not perfect, so we don't want to claim that this is really universal in the same sense of a statistical mechanics and the AC model, but they look pretty robust exponents across regions. For example, this one is very remarkable. Okay, so it's a scaling variance. It seems very clear that there is a scaling variant, but has it anything to do with criticality in the sense of a phase transition? Okay, to explore that, we take this model, which is the most stupid model that you can think of if you want to model neurons. So it's the linear rate model. It's very broadly used in neuroscience, and it simply tells you, I mean, in terms of physics, this is just okay, let me tell you in terms of neuroscience. X is the activity of neuron I, and it's changing in T, some time scale. And what it does is it just decays. So if the neuron is not talking to anyone, it just switches off, goes to 0, the rate of firing. But it's coupled to other neurons, and there is a matrix telling you the influence of another neuron J on Y. And we take this matrix in the simplest possible study just to be a random matrix. So we take Gaussian distributed values, positive, negatives, whatever, with some thickness, with some dispersion and 0 mean, right? And we put some noise. So in this very simple model, just changing the global coupling or the width of this distribution, you know that the distribution of eigenvalues of the random matrix obeys this law, this circle law. And when this is broad enough, the bluish here, then it touches the edge of instability, which is at j equals 1. Why is 1? Because here we have a -1 by convention. So when the largest eigenvalue of this guy is larger than 1, the dynamics becomes unstable. Okay, so the unstable, as is linear, just explodes, but we have a stable regime, critical point, unstable regime. We have that. In this simple model, we can compute very easily, analytically, the covariance matrix, and it's related to omega like this. You have a very simple mathematical relation. And what you would like to do now is to infer using your model. Let me go ahead. Oh, I went backwards. What we'd like to do is to infer what's this matrix omega from measured covariances, right? If we measure covariances, and I was able to infer this, then I would know exactly what's the dynamical state of the network, if it's far from criticality, beyond criticality, whatever, right? The thing is that this is very hard problem. It's the inverse easy problem. You have to compute all the elements of the matrix, all the interactions from all the correlations. That's very, very difficult. I mean, that's not feasible. You would need an infinite number of data, and that cannot be done. But luckily, we have two methods. based relying on theory, on the theory of disordered systems actually, that allow you to infer not the whole matrix omega, this omega big, but you can infer the largest eigenvalue. You can do that. That's just one number, and that you can do with the covariance matrix. So I just very briefly tell you about these methods, very, very briefly. I don't want to enter much into the details. This one I like very much, was proposed by Son Polinsky and his collaborator Yu Hu. And what they do is they take this model that I consider, and they were able to compute the spectrum of the covariance matrix. So they use this formula I put you before, and they can compute as a function of the spectral radius of the width of this matrix. They can construct the spectrum of the covariance matrix. If you want, is this. If you give me the spectrum of this, maybe I can compute the spectrum of this, right? That's what they do. The calculation is not too difficult. And what you see is that the distribution becomes a power law with this exponent here when you are at the edge of instability, right? So when you are at the edge of instability, this is the distribution of eigenvalues of this model. And if you tune it to be close to instability, you have this power law, very nice, which is not the Marchenko-Pastur distribution that you see for random things. So it's not random, it's highly correlated, okay, with this exponent, right? So what you do now is that you take your data and from the actual brain, and you fit them to this distribution. Here I just plot the critical point, but you also have the distribution as a function of the distance to the critical point. So when you are far from critical point, you have cutoffs, right? So you take your data from the brain, you compare with this distribution, and you can infer the distance to criticality. Okay. The second method I have not explained here, but it's also based on the on some properties of the covariance matrix, actually in the dispersion of values. I'll skip it to make it simpler. So we have these two methods. The funny thing is that using them that are completely different, they rely on different mathematical properties and theoretical approaches. We have an estimation of the distance to criticality. This G is like the spectral radius. So if it's 1, it's critical. If it's 0, it's non-critical. And we get a bunch of results that are along the diagonal, saying that the two methods are consistent. So we are getting nice estimations of the distance to criticality. And here are the results. This is what I would like you to look. So this is the largest eigenvalue. So 1 is the edge of instability. And these are the 16 regions we analyzed. So what we see is that all regions are within a 3–4% of distance to criticality. They are all very close to becoming unstable. The dynamics is very rich, it's very critical-like, right? Some regions are closer and some others are farther away. This is something we still do not fully understand. We are working on that. This is work in progress. But we have found this analogy. This is a hierarchy of the organization of the brain in the mouse. And there are regions that have a low score, meaning that they are more sensory. And then as you go up in this hierarchy of information processing, you get to higher regions where you have a higher abstract information processing coordination regions, right? And what we see from our data plot here, this is the distance to criticality. This is this score in the higher hierarchy. And we see that sensory regions are more critical, are closer to 1, and higher order regions in the scale are less critical. And this is interesting. Maybe regions that are critical needs to be very responsive. Maybe that's why they are critical. But this does not fit with other findings by other people. So this is work in progress. I don't insist very much on this. This is unpublished, and I won't insist very, very much. Okay. So just for you not to get lost, we proved first that there is a scale invariance with the renormalization group. Then we proved that the dynamics are actually very close to an edge of instability with models. And now we would like to close the loop and do what? We would like to take this model, again, the very simple model, and generate data, synthetic data with them, time series, and apply the renormalization group, right? because we have the technology to do this phenomenological renormalization group. So we do that. And we do that as a function of how critical is the matrix, how critical is the dynamics. So if the dynamics is very, very stable, look, for example, this is the variance. The variance is scaled like the neurons are uncorrelated. So it's like this is the central limit result. They are uncorrelated. But as you approach the edge of instability, G1, which is the critical point, they become correlated, and amazingly, this variance is scaled with an exponent that in our very simple model, this is the simplest, most stupid model you can imagine, we reproduce the result, the critical exponent we see in the brain of the mouse, this 1.38 with error bars. For me, this was really amazing. I was not really expecting at all that this very simple model could capture this, but it does. Maybe the model is not perfect because when we look the spectrum of the covariance matrix, we see scaling, but it's not perfect. We kind of get the same exponent, but the collapse is not marvelous. So maybe something is still missing from our model, but the main things are there. Good. So I answered these questions, and I now moved to the final part of the talk, another 10 minutes or so, I hope, 10, 12 minutes, in which I would like to connect with the problem of information processing. So actually, to be honest with you, even if I wrote this review paper on the functional advantage of criticality in many contexts, I was never 100% convinced, sure of the real utility of this criticality in information processing systems for various reasons. And I think I myself became convinced after the research that I am going to show you now. Okay, so I will be talking about representations. And for that, I give you this sketch. So we all have some reality where we move like this friend of us, this monkey, and the monkey with the senses is looking this reality. And this is transformed in activity of the neurons in their cortex, right? And now if we look at this activity, maybe we do principal component analysis or some more sophisticated techniques, and we see that some locations in the space of activity correspond to palm tree, some locations in the space of activity is a very high dimensional space of activity, obviously. You have millions of neurons, so this is a million dimensional space of states. But the idea is that maybe each of these inputs is represented, hence the word representation, is represented with a different set of activations of neurons, right? I give you this beautiful example, which is almost too nice to be true, in which you have this experimental result. You have this mouse moving in a logarithm somewhere in the lab, and you want to codify the angle with the direction in which he's moving or turning, right? So if he was moving there and then turns, he does something like that. So you look, I mean, you, the neuroscientists, colleagues, look inside the activity of the neurons in the motor cortex area, and they project this activity, which is extremely high dimensional. Maybe they have thousands of neurons. They project in 3 principal components, and they have these points. It's like a Moebius band, but the important thing is the one-dimensional object. So in this space, which is a projection, I insist, of a 1,000 dimensional space, when you have points like this, means that the mouse is moving in this orange direction. And when you have activity in this location, means that it's moving in this other direction. So this variable angle is codified in a low dimensional manifold within the high dimensional space of states of the brain. This is very, very nice, amazing, I could say, but usually it's not that simple. Usually the things you have to codify is not just something which is one-dimensional, an angle. It's much complicated than that. And here comes this beautiful paper by the group of Kenneth Harris in London. It's already 5 years old, in which they derive a mathematical theorem in which they say that if you want to construct a neural representation on some object that it's very complicated, that has some large dimension by itself, the data, the real world has very large dimension, then maybe I have it better in the, okay, then you have to look at the spectrum of the covariance matrix of the neurons. If the spectrum falls very abruptly, that means that you have a very few principal directions and you have something like the thing I illustrated to you previously. And that is good if you have some low dimensional object, right? But what happens if you have something that is more and more and more complicated? Then you need to put more modes, meaning that this slope has to become smaller. So the spectrum has to become flatter. You start getting more modes, and this thing start becoming more like this. But if it's too flat, it's like this is just noise. This is white noise, right? It's something like that. So the theorem, they prove a mathematical theorem, as I write here. They said, we want to impose that in this very high dimensional space, the manifold encoding the information, we want it to be continuous and differentiable. Why? This is the mathematical condition to impose that if you have two guys like these two here, you want to represent them nearby. You don't want to represent this with a set of activity in your brain and this with a completely different set of activity in the brain. Okay, so if you impose this condition of continuity and differentiability of your manifold, you get that the exponent of the spectrum of the covariance matrix has to be just above 1. This d is the dimension of the inputs. So if you are looking something very complex with a very high dimension in the real world, this number is large, so this is small, and this is a number just slightly above 1. And this is a theorem, right? By the way, when you do in artificial intelligence, they do this thing that's called adversarial perturbation, in which you have that your machine has been trained and identifies this as a cat. But then you put a perturbation that they know how to design, and then the machine gets absolutely crazy and thinks this is maybe a dog, just because the representation is not continuous and differentiable. So you put the perturbation and you end up in a completely different conclusion. Your representations to be continuous and differentiable, right? And the idea behind, again, maybe I'm repeating myself, is that these optimal representations represent an optimal trade-off between keeping as much information as possible so the spectrum becomes flat, but not too much flat, because if it's too flat, you put too much weight onto fine scale details, onto noise. And so the summary is you need a power law with a slope close to 1, above 1. Now the amazing thing, this is really amazing, I will not be tired of repeating this, is that they went to look inside the brain of a mouse, and they showed a lot of images, natural images from the real world to the mouse, like this much images. And while the mouse was looking these images, they computed the activity of 10,000 neurons. They computed the variance of these neurons, the covariance among pairs. They took the matrix, they diagonalized the matrix, they plot it, and voila, they get 1.04, while the theory of optimal representations, mathematical theory, tells you that this should be the exponent. So it's perfect and amazing. And if they repeat it with different rats, with different mouses, and it works perfectly fine. So the mouse of the brain is encapsulating information in the optimal way from a mathematical point of view, right? Again, here is the summary of what I just told you. Of course, if instead of these complicated images, you show much simpler images, then the exponent is larger, because so the slope is more pronounced, because with less principal components, you can encapture the information. And you also see this in the experiments, in the actual mouse brain, right? And this is just new results with 1 million neurons. Again, they say this power law with the same slope. This very, very recent paper confirmed it again. So just to finish, I show you what we tried to apply this to artificial intelligence and to see how it works. And for that, I will not consider this type of architecture, which are the ones we are used to, deep neural networks. I will be focused more on recurrent neural networks, in which the central layer, the neurons are connected with each other. So you have feedback between these guys. So actually, well, and we have the Nobel Prize winners this year awarded to these people. This would be more like Hopfield model, and this could be more like Boltzmann machine, right? Type of architecture, the Hinton. Okay, so again, together with Guille Barrios, my PhD student, we did the following. We consider this prototype of machine learning that maybe it's familiar for some of you, maybe it's not familiar for some of you, but it's not very complicated. It's called, the paradigm is reservoir computing. So you take a network here of neurons, artificial neurons, or virtual neurons in this case, you put some recurrent matrix between them, okay? And now you put an input. The input we are going to put is exactly the same as in the experiments of Harris with images. Okay, in this setup, the funny thing is that you show many images and you do not have to train the network, like in the Hopfield model. You do not have to train these connections to identify or to learn to distinguish that this is a bobcat. What you train is just the readouts, right? So the philosophy of this model of reservoir computing is that you put some perturbation on the network. The network reacts. And then from reading the reaction of the neurons, you can identify what was the perturbation. So that's where the name reservoir is coming from. So imagine you have a lake, a reservoir, you draw some stones, some places, and then some other guy, your friend from the other side of the reservoir, just looking the propagation of waves is able to tell you how you perturb it, right? Okay, so that's how this thing works. More specifically, the model is very, very simple. The state of activity of a neuron at time t+1 depends on the state of all the neurons at previous time. And this is the matrix of connections that we put randomly, very similar to the model I put you before, just a model of random connections. And this is the input that's arriving, and this U is codifying the input with some weight, right? We made something very, very simple with just 2,000 neurons, etc., etc. Key point is that I am going to change this connectivity matrix to make it more stable or less stable. So I can tune the level of criticality of my reservoir. I control this with one parameter. So when I show you the images, what I do is that I put my reservoir to be close to critical. And look, when I show you the images, this is the spectrum of the image. And here I'm plotting the empirical results in the visual cortex of the mouse is the yellowish. And this is the internal representation. So the spectrum of the covariance matrix of the neurons in my reservoir. When I do the same thing, I show the same image. It's amazingly the same. So our model, when I tune it to be close to critical, reproduces the properties of representation in the mouse, in the actual brain. And similar things when I put something lower dimensional. So, okay, so what about performance? So now, instead of the images, that the only goal was just to represent them, we did not train the network to distinguish all the possible images. That was too complicated, too many images, very complicated. We did something simpler. We just taught our machine to distinguish these numbers. This is the MNIST database, this very standard protocol in machine learning. So we teach the reservoir to learn this as a function of my only control parameter, which is the distance to criticality, right? And this is the performance. You see the performance in blue, the blue points. And you see the best performance, 98%, 99%, is when we are in this greenish area. And I tell you that the critical point is this line here. Why? Because I'm measuring inside the reservoir the Lyapunov exponent, lambda, and I'm actually verifying that here is positive or it's unstable, and here is stable. So with my control parameter, when I tune my control parameter here, I'm at the edge of criticality. And the performance of the machine is the best when I am slightly below the edge of criticality. But the amazing thing, and again, I think this is amazing, is that now I plot the spectrum of the covariance matrix. I diagonalize and I compute the exponent of the covariance matrix. And I see that at criticality, I just get the value, magical value 1. And when I slightly below the critical point, I get the optimal result predicted by Kenneth Harris of what optimal representations should do. So my reservoir, which is very simple, very stupid, with very simple neurons, when I tune it to operate close to critical, is able to compute optimal representations of this database. I think that's beautiful, but this one is even nicer, I think. Now, what I'm going to teach my reservoir is to identify the Lorentz attractor. So this is very typical. This reservoir computing you use to predict time series, and people are very used to do that. So instead of images, now we are plotting time series. So we have some input, then we train the readouts similarly to what we did before. And the protocol is essentially the same, but now we learn this. And again, now I have two parameters, but again, if you just look in this column, you see that when the maximum Lyapunov exponent crosses, so this is the critical point, the performance is optimal. This is the further predicted point is optimal, the error is minimal, and again, amazingly, the representation exponent becomes close to what it should. Now it's not 1, because the dimension of the Lorentz attractor is, how much is it? 2.1. So this is 1.95, and indeed I get 1.95. So the optimal is not just 1, it depends on the dimension of the input. But in this case, the nice thing is that I can look inside the principal components to see what my reservoir is doing, plot the principal components as a function of the distance to criticality. So if I am very stable, the reservoir is really codifying in a 2000, my reservoir has 2000 neurons. So in a 2000 dimensional space, when I project it in the right way, I do a dimensionality reduction, I see that inside my reservoir, it's codifying the Lorentz attractor. But this is not optimal. Here the performance is very bad because it's just following what it has done. It cannot generalize. Instead, when you're close to the edge of instability, it becomes less smooth, but here is where it performs better, when it's able to predict. And if you put some modification, some perturbation, it always predicts. Instead, if you go beyond the edge of instability, then the attractor becomes peaky. It's not continuous, it's not differentiable anymore. So we reproduce beautifully, I think, the theory of Kenneth Harris, that the attractor, the representation has to be smooth, differentiable, but as complicated as possible to accommodate for as much information as possible. And of course, when you're here, the spectrum is what you expect to be the optimal one. So just to close, the final idea is to use this not to run codes in silico, but to develop actual neurochips. So this is a collaboration that I got involved with the people in Heidelberg and Göttingen, and they have actually constructed this chip that emulates a situation similar to what I told you before, this reservoir machine. They train it in different ways, and the idea is to see if it works better when you go to criticality. Nicely, this thing, when it works, consumes 10,000 less energy than a conventional architecture and is 10,000 faster. But we are not still there. It's work in progress. I would love to tell you that we have this implemented in a chip, but we have not yet. And even more science fiction-like, together with our collaborators, Jordi Soriano and Hideaki Yamamoto in Japan, they actually construct real neurons with real alive neurons. They put in Petri dish, and we put them in nice architectures, and we try to design connections, and we try to do reservoir computing employing this thing. But again, this is a dream that maybe one day comes true, but for the time it's just work in progress. So my summary, I hope I got not too long, is this. Operating near the edge of a transition, a phase transition, near criticality, near the edge of instability, is an excellent solution to perform generic computations collectively, because look that this reservoir computing is generic. You can do, I have learned to recognize images, numbers, time series with the same machine, right? So it's generic computations. And evolution has certainly found and exploited this idea. And we, humans, we are starting to leverage it for artificial intelligence. And very important, I have to thank, well, my fundings, but especially Guillermo Barrios Morales, who has done most of the analysis that I presented here, but also Serena and my new PhD students who are working in some of the other projects that I also discussed marginally here. And that's it. So I'm ready to take questions if you want.
Thank you. Thank you, Miguel, for this amazing talk, and with so many inputs and so many interesting topics and amazing results. So we are ready for questions. So are there any questions from the audience? Everybody is frozen from the for the result. So I have many questions. So I will ask some very basic one, Miguel. So it's really it seems that for example, in the case of where you use a the linear model for the neuron activity. So using a linear model is certainly a good starting point, but of course it is a very simple model. So do you think that, for example, the result about the exponent that is coming out so beautifully from your modeling scheme does depend on the type of model that you use? Is there some such a strong universality that you can use even a linear model to get such an amazing result?
Yeah, the answer is I don't know. I have a new student, master student, and his project is exactly this thing. So he's exploring now this with an hyperbolic tangent and with different type of noises and with different types of matrices, not Gaussian, but because the real connections in the brain are not Gaussian matrices. For example, you have excitatory and inhibitory neurons, so the architecture is very different, and heterogeneity, so the distribution is not Gaussian of weights, is log normal. So we are putting all these ingredients one at the time to see which ones are really important and which ones are not, and if you can change universality or not. So this is work in progress. Yeah.
So what is your explanation why the exponent is coming out right with such a simple model? So it's some kind of
no, to be honest,
no.
I have any good idea. I mean, I was absolutely surprised when I saw just see this scaling. I mean, the fact that scales,
amazing.
Expecting that because it's critical, so I was expecting scaling variance, okay, but maybe it's a coincidence, could be a coincidence, lucky coincidence, but it's two coincidences because also the Z is the same and also this is 0.78, so it's really, I don't know if we were just lucky or not, but that's why I tell this is work in progress.
Okay. So are there other questions from so there is one question from Valeria D'Andrea. So please, Valeria, you can switch off.
Yeah. Hi. Hello. So I don't know if you can hear me or see me.
Yes.
Hi. Hi.
Both.
Sorry. Thank you for the amazing talk. I have just a simple question. When you apply the reservoir, you just change it. You maintain a random connection in the matrix, right? What do you think that it could happen by changing, the topology of the network? Do you think that there could be some effect on the performance or other?
Yeah. There are some people, including ourselves, with Anna Levina and Victor Buendia, studying how the architecture of the reservoir can improve the performance. So, for example, now we are constructing reservoirs that are kind of fractal. So they have some construction in which you have moduli within moduli. So when you have something very complex to do, you have the possibility to delocalize the representation in different parts. So yeah, the question is this work in progress. There are people studying these type of questions, yes. Also, there are people studying synaptic rules. So you can put plasticity inside and it also changes the performance. We are also doing something in that direction.
Okay. Thanks.
Thank you.
Are there other questions?
Perhaps I have one.
I recognize the voice is Stefano. Stefano, please.
Concerning the reservoir computing applied to the Lorentz model, I missed what is exactly the Lyapunov exponent you are referring to, because usually when you reconstruct the exponents with reservoir computer, you compute all the exponents of the reservoir, and the first three, say, should reproduce the Lorentz one. So which is the exponents you are referring to?
Deleted the maximal Lyapunov exponent here. This is just
of the reservoir.
Of the reservoir, yeah.
But this should be positive anyhow, no?
No. Look, I'm not looking at the activity. I'm not looking at the Lyapunov exponent of the time series. The time series enters here, and this reservoir just follows the dynamics, which is this. So this is the input. This is the Lorentz attractor. You have some weights here that also are random. Then you have your reservoir with some random connections, and these guys that go beyond between -1 or 1 or 0 or 1. So there is some dynamic here, and the dynamics is mostly tuned by two things: how strong you couple with the input, this epsilon, and the spectral radius of this. So how critical this is. But as you have two different contributions, inputs plus intrinsic, then the best thing you can do is actually compute the maximum Lyapunov exponent here and see where it changes sign. Okay. And when it changes sign is when you have that the optimal representation is here and the optimal performance is here.
So this is the Lyapunov exponent of
the maximum Lyapunov exponent, yeah.
Is there of the reservoir without input when it's running in autonomous mode, as they say?
No. It's with the input. It's all together. So yeah. This is a very delicate issue that you say you have to look for critical or more unstable, but considering the input. Okay.
Okay.
So it's not an autonomous system. That's why it's not enough just to tell you what's the spectral radius of this.
Okay.
Right.
So it's different from the standard thing when you look at the.
Yeah, it's a non-autonomous system. Yeah.
Okay.
So.
Okay. Thank you.
Other questions?
Maybe I can ask a question. Thanks, Miguel. Beautiful talk. I was wondering, your conclusion that is actually convenient to be close to instability, this conclusion, how can you understand this statement considering that an healthy brain realizes the balance of excitation and inhibition?
How do I understand it?
Yes. How is balance of excitation and inhibition related to be close to instability?
Yes.
I mean, there are two different languages, no?
Yeah.
That sometimes the balance of excitation and inhibition is used as a hallmark for the healthy functioning of the brain. How do you reconcile the two statements?
So I would say that critical here means a good balance between excitation and inhibition. If you have too much excitation, you become epileptic-like. If you
sure, this is clear, but concerning being close to instability.
Exactly, but for example, here in Parkinson's disease, many modes of many different frequencies, many different bands, they are very close to instability. So much more in healthy controls. And we know that in Parkinson, there is an imbalance in excitation inhibition. So inhibition damage is not inhibiting enough. So it's getting too excited. It's getting overly excitatory. So overly oscillatory also in this regime. So in my view, excitation inhibition means controlling the distance to criticality. That's, I mean, talking in very simple terms. I don't know if you agree with that or if you want to say something more subtle.
Yeah, no, I was just trying to think in terms of being close to instability in this language. What does it mean? Means that
my
yes,
opinion is close to balance.
Close to balance.
Inhibition dominated.
Inhibition dominated. So slightly subcritical, you would say.
Slightly subcritical, exactly. So you are close to becoming perfectly balanced, but when you're balanced, you become unstable. So you have to be slightly inhibition dominated. And I think that's very broadly acknowledged and appreciated by neuroscientists, that they believe that the standard state of the brain is inhibition dominated, but very, very close to becoming active.
No, I agree also, because if you think in terms of the power spectrum of the activity, if you are precisely at criticality, you have Brown noise. You don't have 1 over F. Exactly. So to have 1 over F, you have to be slightly subcritical.
Right.
Thank you.
Thank you, Lucilla.
We have another question from Lorenzo. You can mute yourself. Okay.
Yes, thank you. I have a question about the linear rate model, because I know that this linear rate model can be trained with the backpropagation through time. We actually do that. That is, I mean, an artificial method to train this kind of model.
To train to do what? Sorry.
For example, a classification task or regression task, like we train this kind of model to solve like the MNIST data set. And I was wondering if are there any results about the criticality of this model after the train? Because I expected that since this backpropagation is not BO, probably the criticality is broken.
Yeah, but when you say you train, you mean that you change the weights.
We change the weights, yes.
Okay. Then I'm pretty sure that when it works fine, probably you are near the edge of instability.
Okay.
But the standard procedure is more the reservoir computing one, but I'm not aware of this one you are mentioning. I believe you. I mean, you can train the weights to do something specific. It's more in the optic of Hopfield model, right?
Okay, yeah. Thank you.
Okay, are there more questions? So okay, since there are no questions, we thank again Miguel for this beautiful seminar. Thank you so much, and
my
good afternoon to everyone.